Hello, so today on Road to 250 we're going to talk about skeletal muscle and conduction and throw in some heart blocks and then have some practice questions at the end. So I'm going to start with the skeletal muscle portion with the structure of a myofibril. So I'm going to break it down to basics here. We have a muscle made up of individual cells called muscle fibers, and within these fibers there are bundles of myofibrils, as we see here. And a myofibril can be subdivided into individual sarcomeres, which we see here. And these are demarked by what is known as Z-lines, or Z-bands in this picture. So this line right here is going to mark your sarcomeres. And they are composed of filaments. We have the actin filament and the myosin filament that interact to have muscle contraction, but we'll get to that in a little bit. When contraction occurs, we have an interaction between these two filaments, and what happens within our sarcomere is that our I band gets shortened, as well as a shortening of the H zone or H band, however you want to call it, zone, band, interchangeable. And there is something called titin that anchors myosin and is important for the elasticity of striated muscle. So to go a little bit more in depth, so within those muscle fibers, we have the sarcoplasmic reticulum, and that is going to be this yellow stuff out here, this whole thing, all of these that we see. And they contain T-tubules and terminal cisternae. So T-tubules are extensions of the surface membrane, and that's going to be the inner portion here. I'll clear that up for you. And these are part of the extracellular component. And then we have the terminal cisternae. This picture's a little blurry, sorry about that. This is the internal membrane system, where we have the sarcoplasmic reticulum storing our calcium. So that's this edge guy right here. And in skeletal muscle, which is what we're talking about today, most of the calcium is stored in this terminal cisternae close to the T-tubule system. As you can see, they pretty much look like they're part of the same structure right next to each other. Okay, so within the sarcomere we have functional proteins. We have proteins of the thin filaments and proteins of the thick filaments. And this is going to come up in a question later, so make sure you're paying attention. So the structural protein of the thin filament is actin. And actin has attachment sites for myosin. Of course, they interact with one another. And we have something called tropomyosin that blocks these binding sites on actin. So if you can see right here, we have tropomyosin, like this long little chain going over actin, blocking these binding sites. And then we have troponin, which is composed of 3 subunits. We have troponin T, T standing for the fact that it binds to the tropomyosin. We have troponin I, which binds to actin and inhibits contraction, and then we have troponin C, which binds to calcium. So you remember what they bind to by their letters here. So at rest, no calcium is being bound, and actin and myosin won't interact. All of the binding sites are still blocked. There's nothing going on. We're at rest. When calcium does bind, so you can see here, the troponin-tropomyosin complex, this whole thing, moves out of the way to expose these myosin binding sites so myosin can bind, forming cross bridges, and we can have contraction and have that occur. So that's the thin filaments, and on the thick filament side, we are referring to myosin. Myosin has ATPase activity, and that interaction with ATP. So when ATP gets split by this ATPase, it puts myosin in a high energy state and increases myosin's affinity for actin. So I mentioned that the binding sites have been exposed. So once they're exposed, myosin can bind to actin, and then the energy formed in that what's the word I want to use? In that interaction causes the chemical energy to transfer to mechanical energy, where myosin gets to pull the actin filament and create tension in the muscle, and this is what's referred to as the power stroke. So if there is enough force to move the load, and the muscle can shorten, and that's referred to isotonic contraction. But if there's not enough force able to shorten the actual muscle, then it's isometric contraction. So throwing that out there, some terms for you guys in case they come up. So a little bit more about these cross-bridge interactions. So like I said before, the whole cycle begins when free calcium is available and can attach to the troponin C, which moves the tropomyosin so that myosin can bind to actin. And then contraction occurs when there's a continuous cycling of these cross bridges going on. So your muscle is continually going through these shortenings that are forming because of these cross bridges. So where ATP comes in is that it's not required to form the cross bridge, but it is required to break the link with actin so that another cross bridge can be formed afterwards. So that's where you get the continuous cycling going on. And the contraction, again, can also continue until there is a withdrawal of calcium, at which point cycling will stop here because our tropomyosin is back to covering our myosin binding sites on actin, so they can't bind, we can't form those cross bridges. Or if we run out of ATP, at which point we are stuck with our myosin bound to actin and it can't let go. And we see this in rigor mortis and wouldn't normally occur under physiological conditions in alive people, so keep that in mind there. So like I've mentioned before, calcium is pretty important for this whole cross-bridge cycling contraction stuff to happen. And the sarcoplasmic reticulum that we talked about has a very high concentration of calcium. So there is a very strong gradient for calcium to diffuse into the cytosol. So there's two receptors that are involved for this movement of calcium from the SR into the cytosol. They are the dihydropyridine receptor and the ryanodine receptor. So the dihydropyridine receptor, as we see here, is voltage gated, and it's located on the sarcolemmal membrane, as you see here. This functions as a voltage sensor in skeletal muscle. So in skeletal muscle, when it's at rest, this receptor functions to block our ryanodine receptors, which I will talk about. So our ryanodine receptors are our calcium channels on the sarcoplasmic reticulum membrane here. Do that a little crappily. So when the muscle is at rest, like I said, these ryanodine receptors are blocked by our dihydropyridines, as you can see here. So calcium cannot leave out into the cytosol. This can't happen. Make sense?
Yep.
Okay. So during our actual contraction process, when there's an action potential going on, I'm going to go through the sequence of what occurs. So we have an action potential initiated at a neuromuscular junction. So see a neuromuscular junction going on. We have our action potential created. So this travels down our T tubules, and that will cause a conformation shift in our dihydropyridine receptors. So these guys get a shape change. And what that does is move it away from blocking the ryanodine receptors. This space is now open for business. So calcium can now leave out into the cytosol. Because we have so much calcium in here, it's just going to follow the concentration gradient and just diffuse out. And with calcium exiting out, this is also going to stimulate the ryanodine receptors, more of them, to open up and continue to release even more calcium. And this is what is called calcium-induced calcium release. And then this calcium is available to bind to troponin C, which moves a tropomyosin, allowing myosin to bind to actin, and then we have our whole cross-bridge cycling going on, tension happening, and contraction occurring. After that, calcium is pumped back into the sarcoplasmic reticulum by something called calcium ATPase. And that is on the SR membrane, and it is called SERCA, or sarcoplasmic endoplasmic reticulum calcium ATPase is the full name, but we'd like to be short and abbreviated, so SERCA is easier to say and remember. So when calcium is being brought back into the SR, it's no longer bound to troponin C, so tropomyosin can cover up those binding sites again, allowing the muscle to relax, and so on and so forth. It can continue on in this cycle. And yeah, and this does require ATP to dissociate actin and myosin, so that is also required for the muscle to relax. And this is from first aid. So just to point out some differences, we're primarily talking about skeletal muscle today, but when we want to differentiate skeletal from cardiac, our skeletal muscles have one T-tubule and two terminal cisternae, versus the cardiac having just one of each. So skeletal muscles have a triad, while cardiac muscles have a dyad, but you do want that extra sarcoplasmic reticulum energy calcium stores coming in there. So just to go through this conduction system one more time, we have an action potential opening our voltage-gated calcium channels, allowing the calcium to come out into the cytoplasm. And these are our dihydropyridine channels. We have muscle cell depolarization in the motor end plate. And that depolarization travels along the T-tubule. And then we have our voltage, like I said, our dihydropyridine receptors, which have a conformational change, causing calcium release from the SR. And then this calcium can bind to troponin C. That moves tropomyosin so that myosin can bind to actin. Put that out there. And myosin also releases bound ADP and additional phosphate, which displaces myosin from actin. So this is where ATP comes in to separate the myosin from the actin. And this is where we have our power stroke occurring. And again, if you guys can kind of see here, we have our H bands and our I bands. These are the ones that are going to shorten during a contraction. And first aid mentions his shrinkage to kind of help remember which of these will be affected with contraction. So those two. And finally, binding of a new ATP molecule causes detachment of myosin from the actin, and myosin can go back to having its high energy position or cocked position for our next contraction cycle. And in questions, we'll often see images like these, this guy right here, that will ask about certain portions of the sarcomere, which we have a couple of examples later on. So just make sure you know we have our H bands and our I bands. Our H bands demarked by our M line, which I like to remember as sort of like the middle of the sarcomere, and also where we find our myosin, and then the Z line being the ends, like kind of like the end of an alphabet. You have the end of the sarcomere being Z. Alrighty, so any questions about skeletal muscle and contraction before we move on to heart blocks? Okay, so when I'm talking about heart blocks, I'm talking about our atrioventricular blocks. This is where there are conduction abnormalities of the AV node, where there's some sort of interruption leading to a prolonged PR interval. So that's kind of characteristic of our AV blocks, is a prolonged PR interval across the board. So in first degree heart block, like I said, we have a long PR interval, so greater than 200 milliseconds, where there is, like I said, slowed conduction through the AV node, but rate and rhythm are typically normal. So this is a benign and asymptomatic condition. It doesn't necessarily need treatment. We're just having a longer differentiation between the atrial conduction and the ventricular conduction. And a helpful little poem phrase to remember first degree is, if the R is far from P, then you have first degree. So if that helps anyone, that's on there. Then we have second degree heart block, which has two different types. In second degree heart block, we do have, so with every QRS complex, there is a P wave in front of it, but not every P wave has a QRS complex after. So if that kind of makes sense, here we have a P wave but no QRS, this guy occurring here, but where there are QRS waves going on, there is a P wave occurring before it. What this basically is telling us is that some impulses are not transmitted through the AV node. It's not making it all the way through. So like I said, there are two types. There is Mobitz type 1 and Mobitz type 2. So Mobitz type 1 is also known as Wenckebach. And here we have a progressive prolongation of the PR interval. So it's getting longer and longer and longer until a ventricular beat is missed, and then the cycle begins again. So we have an unsteady rhythm. So as you can see, we have a longer PR interval until there is no QRS wave at all, and then it starts all over again. And this one is also usually asymptomatic, and the intervals between the R's are variable, but there is a pattern, as you can see. So regularly irregular is another way this is referred to. And going back to our heart block poem, this portion you can remember with the phrase longer, longer, longer drop, then you have Wenckebach. Keep that in there. Then we have Mobitz type 2, where the PR interval is constant, so it's not lengthening. But we still have those drops going through. So the rhythm here can be steady or unsteady depending on the ratio of P's to QRS's occurring. So again, there are drop beats, but there is no change in the length of the PR interval. Whereas in type 1, we do see a longer, a progressive lengthening of that interval. And this particular condition is often treated with a pacemaker because it is a little more serious, because these beats are random. It's not regular. Something is causing the electricity to fizzle out in the bundle of His and not be able to move on into the Purkinje fibers for the QRS complex to do its thing. And this is usually caused by fibrotic changes in the bundle of His, so something more serious is going on. And the poem line for this guy is, if some P's don't get through, then you have Mobitz 2. So again, this one is random, so keep that in mind. And last we have third degree, where there is a complete dissociation of P waves and QRS complexes, impulses not being transmitted through the AV node. What this basically means is that the atria and the ventricles are not interacting with each other. They beat independently of one another. The rhythm is usually steady, and the heart rate is usually very slow in the ventricles. The PR intervals aren't exactly consistent. And again, like I said, they're not transmitted through the AV nodes, so the rate for the P waves is going to be different than the rate for the R waves. So we have the atria firing at one rate and the ventricles firing at its own separate rate, not related to each other. And this is usually treated with a pacemaker as well. And a common cause of third-degree heart block includes Lyme disease. Can anyone tell me the offending organism for Lyme disease?
Borrelia burgdorferi.
Right. So what is happening in Lyme disease that is causing this heart block?
Carditis?
Yeah, kind of. So the spirochetes are gonna spread throughout the blood and enter the heart and affect or activate the immune system within the heart. So you have a local inflammatory response, which results in fibrosis and damage of cardiac tissue, which is why we have a messed up conduction going on. And the last line of that heart block poem here is, if P's and Q's don't agree, then you have third degree. And yeah. So any questions about that before I go on to quiz you guys?
With the type or the third degree, it's also blockage of the Purkinje system, right?
Yes.
Thank you.
Mm-hmm. Any other questions? Okay. I'm going to start with this guy. We have an 8-year-old boy is evaluated in the office due to difficulty walking. Patient has problems running, jumping, and walking up steps. There is no history of neurological disorders in the family. Physical exam reveals weakness of lower extremity muscles bilaterally. Electron microscopy of a biopsy sample taken from the calf muscle is shown. Which of the following labeled regions contains only thick filaments and no thin filaments?
B. Yeah, I second that one.
Yep. It is actually E.
No, it's E. It's E. Yeah.
Well,
sorry, my mic was muted.
Oh shoot, that's
It's a little hard to tell that.
Yeah,
they look all the same.
C is included in B.
Oh God, come on.
And C is where we're having actin and myosin overlap one another. So.
Dang it.
Yeah. It's okay. A little
I
hard
believe
to
your
see.
picture.
It's not my picture. It's just the picture. Okay, we'll try again with a different question. A researcher is investigating the structure of the sarcomere using skeletal muscle obtained from an experimental animal. He develops... Oh, actually, I think I'm using the wrong picture, but I'll fix it in 1 second. Okay. He develops monoclonal antibodies directed against a specific skeletal muscle protein and finds that these antibodies disrupt the binding of actin to structural support elements within the sarcomere. Electron microscopy of the sarcomere is shown. Which of the following labeled regions do these antibodies most likely bind? And before you answer, let me just fix this real quick because I forgot to use the correct picture. That is going to be D. This is going to be E. Sorry, guys. And this is going to be F. Okay. Now you can go nuts. Binding of actin to structural support elements.
A.
Okay. Anybody else?
Are you talking about Titan?
See?
Let's see. And yes, to your Titan question.
Why? Actin to structural support elements.
Okay.
I almost feel like it's D now.
Yeah, I'm going with D. Sorry, I changed my answer.
But Titan is the anchor. D is the interlocker.
But isn't D thin filaments?
No, D is where they overlap. A is the Z, or not the Z. A is the whatever the fuck it is. C is the Z line. A is where actin anchors. C is where myosin anchors, and then D is where they overlap. So disrupt the binding of actin to the structural support. Whatever, fuck it, I'm sticking with A.
Okay.
I don't know. I have no idea.
A is correct.
No.
So A is our Z line. So this
Oh, that's the Z line. Oh.
is our sarcomere.
So
this-
what's that line called? That's the I band?
This whole thing is the I band. So if I
But what's the thing you just drew the little line on?
Is that the M band, the middle one?
this is the M band
M for myosin. Goddamn.
or M line, M band, whatever. And let me change these again.
So
That's D.
So wouldn't D be thin filaments?
D is where actin and myosin interact, so that's both thin and thick filaments together.
Damn.
And A is where the Z line is, which is the end of the sarcomere, and that's where actin is binding to structural support elements and not to myosin, which is where we see D. Sorry for using the wrong picture, but...
So wait, what is E then?
E is, what is E? E is the I band, so that's where we have just the actin. So the darker portion, all of this here, is where myosin is. And all of this lighter portion is where actin is. And where they overlap is D. And then these are the separations for each sarcomere.
Okay.
Okay. And last question here. Okay. A 67-year-old man is brought to the emergency department by his son after a syncopal episode. The son was helping his father clean out his garage when his father complained of dizziness. As his son was helping him into a chair, the patient lost consciousness. He woke up spontaneously about a minute later without any disorientation or confusion. An electrocardiogram demonstrates bradycardia with regular rhythm and narrow QRS complexes. However, there is complete desynchronization between the P waves and QRS complexes. Which of the following locations is most likely responsible for pacing this patient's ventricles?
AV node.
Okay.
Yep.
AV node is correct. So
1976 for some reason.
that's the QID number in case anyone's interested for you.
Oh, for first aid? Oh, for you.
UWorld, yeah. So, well, yeah, pretty self-explanatory there. SA node is responsible for pacing our atria, and our AV node responsible for pacing our ventricles, and everything else is pretty self-explanatory.
Well, I just remember that the AV node is kind of like the communication between the atria and the ventricles. So if you have a miscommunication in the AV node, then you're not going to have the synchronized contractions.
True. So with this particular line, complete desynchronization between the P waves and QRS complexes, a little additional question: what condition does this patient have?
Complete heart block?
Right. Complete heart block, also known as...
Wait, what is it? If the PR and the P don't agree, then you have type 3?
If the P's and Q's don't agree, then you have third degree.
Oh. Close enough. There's a 3 in there. Whatever, I've never heard those before. Are those in first aid?
They're not.
Okay, I was going to say, I've never seen that. So.
I added them to my first aid, but no, they're not in here.
Okay. I was asking. I've never seen those before. They were cute.
Divya, did you write them?
No, I got them. So when I was in physio, we had to like present different things, and someone was presenting heart blocks. They did the heart block poem. Like you can Google it. Oh yeah, that's all I have for you guys.
So that last question, is that patient representing an AV dissociation?
Yeah.
Okay.
Yeah, and their ECG would be just all over the damn place.
Yeah, because the ventricle, yeah, okay.
You would just have free-firing ventricles that are not associated at all with the... Sorry, you'd have QRS complexes that are not associated with the PR intervals at all.
Yeah, they have different rates between the two.
You'd see PR, PQRS, QQR just randomly all over the place. So, and surprisingly enough, when I was doing the cardio section of step 2 questions, I was expecting a lot more of like, oh, you need to know how to read an EKG, and like I was like on deck to read that, the only EKG book you'll ever need, because I have that on my on my tablet, and I was ready to read that shit just because I thought that's what the question would be. Nope. There's like 2.
Step 3 has them.
Out of 300, there's like 2. Oh yeah, I would imagine for step 3, but like pretty much the amount that you need for step 1 is what you need for step 2. They're more interested about the drugs.
Yeah, I had a really hard time trying to find a heart block question in the first place, and that was like the closest thing I could find on UWorld.